meeting is being recorded.
So whenever you're ready. Okay. It's a pleasure to introduce Dr. Shiladitya Banerjee. He is an assistant professor of physics at the Carnegie Mellon University, and he works in the area of theoretical and computational biophysics. Dr. Banerjee, before moving to Carnegie Mellon a couple of years back, he was a Royal Society University Research Fellow. He was on the faculty of the University College London, where he joined in 2016. Before that, he was a long-term postdoc at the University of Chicago, and he did his PhD from Syracuse University, and I believe his thesis was awarded the best thesis award in the United States. But more importantly for us, before going to Syracuse, he was an undergraduate student at the Chennai Mathematical Institute. So it makes us doubly happy to invite him to present his talk today on the physics of living and evolving matter. Shiladitya.
Yeah. Thank you very much, Sridhar. I'm very happy to be here, and thank you for the invitation to speak. Yeah, it's always good to come back to CMI and give talk. And yeah, I mean, I owe a lot of my academic achievements to the education I received at CMI. So my talk title was pretty broad, and I tried to narrow the focus here. And I realized that I'm going to be talking about biological systems and mathematical and physical modeling of living systems, which I think haven't been touched upon so far in this series of talks. So I'm going to introduce the topic from a very mathematical standpoint, and all the math and the physics will be fairly simple, but there will be a lot of surprises, okay, which I'd like you to appreciate. So my talk today will be specifically focused on how living systems grow, replicate, and control their shapes, right? That's what morphogenesis means. How do living systems acquire their shapes? And what you see here is a time lapse of a bacterial cell, right? A bacteria is tiny microorganisms, which can be pathogenic, or and it's also a nice model for physicists to study living system, because it's the simplest cell, right? It's a unicellular organism that has a specific life cycle. It grows and divides, right? And gives birth to new cells. So this process goes on and on, right? Replication. So so we would like to understand what's underlying this, right? So how can we mathematically model such systems? And is there new physics in these systems, right? So at any point during the talk, if you have any questions, please feel free to stop me and ask it, okay? So let me start by very basic. So here you see a movie of bacterial cells growing and replicating, right? So the first question as a physicist I'd like you to ask, like, what controls bacterial growth rate, right? So looking at this movie, I can measure how fast the systems grow, right? So here's a population of E. coli cells that are growing, okay, when you feed them nutrients, right? And they grow exponentially, okay? So you can mathematically model the population growth, okay? And initially they grow at an exponential phase, right? So that means that the rate of change of the number of the cells grow in proportion to the number of cells. As time goes on, the sort of food runs out, the nutrient runs out, so the growth stops, okay? So this type of population growth can be modeled by something called logistic growth, okay? So this is what's happening at the population scale, right? But if you measure more variables, right? So if you measure more observables, you find more surprising results. So one of the interesting results is if you measure, for example, the length of the cells, right? So as you can watch that as the bacteria grow, they grow along their length, right? So nothing is really happening in the other dimension. And if you measure the length distribution of bacterial cells at the time of their birth, which is LB, and at the time of the division, you see a fairly tight distribution of the cells. Right? So the probability density is fairly well maintained, right? So that means that there is some mechanism for regulating size, right? So there is not a lot of variability in size. So the fluctuation is about 10%, right? So so that's pretty interesting. So how do we get such a tight control of size when there is a lot of stochasticity and noise in the system? So noise is very inherent in biological systems, right? So that's something we'll try to touch on. And the second thing is if you measure the size of the cells, right? So and if you plot it against the rate of growth, right? You find a surprisingly simple exponential relationship, right? Faster the cells grow, bigger they are, right? And so the field kind of you can think about the systems in terms of some macroscopic variables, right? Size, shape, rate of growth, and from experimental data, so you can map their relationship. Now, as a theorist,
this graph, cell size versus growth rate, is this across species or is it for one species of bacteria?
Yeah, that's a very good question. So this is for a single E. coli species. I'm going to later show you data for other species. So this is for E. coli cells. Okay, E. coli cells are commonly found in human gut, and it's like one of the most well-studied bacterial cells. Okay. So as a modeler, right? So as someone who does mathematical modeling, we'd like to ask why this is, right? And to understand this, right, we need to look at the dynamics at the scale of single cells, right? So we'd like to understand how single cells, like single organisms, coordinate growth and division, right? So how fast they grow determines how soon they will divide, right, and go on to the next generation, right? And the coupling between growth and division actually will set size of the cells, right? So how long they will grow will determine the size, okay? So yeah. So about like 10 years ago, people started studying the systems at the at the scale of single cells using microfluidic devices. So what I'm showing here in this movie is a microfluidic device. What they are is like the narrow channels, typically of the width of 2 to 3 microns. So these bacteria are basically, the length is like 2 to 6 microns, that's the variation you can expect, and the width is like 1 micron, right? So, and what you see here is the growth of bacteria. So there is a mother cell at the bottom of this microchannel that grows and divides, and as it replicates, it pushes up the daughter cells. And at the top of the microchannels, you have a fluid, like a shear flow of fluid, right? So which sweeps away any bacteria, right? So you can image this for long periods of time and collect thousands and thousands of data, right? And using this data, you can actually discover new, I would say, physics and models, right? So the first thing I'd like to ask is how does bacteria regulate their size, okay? And how do I actually get a distribution like this, right? So let me first try out a few models, and I'll show you the failure of models, which is, I think, very important, right? When you first try out a model of a system which you don't know anything about, right? You need to try out the simplest possible ones, and then those models fail and you try out something else, right? This is how science progresses. And without knowing much about the biology of the system, but just by looking at the dynamics, yeah, so one important thing is when you look at the, from these experiments, when you measure the length versus time, okay, this is what the trajectory of the system looks like. What I would like you to appreciate is very stochastic, okay? It's not like the cell is doubling in length and then dividing, right? So what I'm showing here is the length curve of one mother cell as you follow it through generations, okay? And you see that, so these red dots are, it's when a new cell is born. And you can see that the timing of the division is pretty random. The size is pretty random, right? you can blow up this at semi-log scale, and you see that the growth is fairly exponential because you see linear lines, right? So when you plot something on semi-log scale, it looks linear, it's exponential, right? So the question is, how is cell size controlled despite stochastic variations, okay? All right, so noise is important, but also there is some sort of deterministic behavior that emerges at the population scale, right? So yeah, so let's first try out a simple model. And the first model I'll call it's a timer model, okay, of how cell size is regulated, right? So the math is pretty simple. I just want to go through it because some surprises are there, right? So in the timer model framework, so the hypothesis of the model is that cell division occurs after a constant time period tau, right? So let's call VB the volume of the cell at birth, right? And as time progresses, the volume increases exponentially, and cell divides after a constant time period tau, and VD is the cell volume at division, right? So one thing you realize is that if you grow for a constant time period and the growth is exponential, that means that you grow as a fixed multiple of your initial size, okay? Because this quantity here is constant, right? So you grow as a fixed multiple. So yeah, so one possibility could be that you grow double your size and then you divide, right? That's what people think intuitively. That's what happens, right? What happens is that after division, the cell splits into two cells in the ratio R, okay, which is called a division ratio, right? So you split the cell into two and you continue this cycle, okay? And for simplicity, I'll assume that R is equal to half because you have symmetric divisions. You can also do similar calculation for asymmetric cell divisions, okay? So here's the important step in the model, right? So tau is the cell size at birth, sorry, cell division time. And I'll add a small noise in the cell division time, okay? And the noise is actually motivated by experimental data where you see variability, right? So I'm adding a simple noise in the time period, okay? So the noise is like Gaussian. So you draw it from a Gaussian distribution. It has zero mean and it has a finite variance, okay? Now let's see what happens with this model as I iterate through generations, right? So first of all, I can compute the volume at birth in generation n+1, depending on the volume at division at generation n, and divide it by half, right? So I get this simple looking relation, right? So I can take a log on both sides because just for mathematical simplicity, convenience, I'm taking a log on both sides. And what you see here, if you write that tau n is log 2 over kappa, as I said in my previous slide, plus some noise, then you add, so what you find is that the log of cell size is log of cell size in the previous generation plus a noise term, okay, kappa eta n, okay, which is the noise. So you can continue this, right? Continue this iteration, and what you can find is that the cell size in generation n or n+1 is some constant plus some addition of noise terms, okay? Which means that now if I take an average, right? Because all the noise has zero mean, right? The average is well defined, the mean cell size is well defined. So the mean cell size is set by the cell size of the first generation, right? Which I call 0 here, okay? So the mean is well defined, so okay. There is no surprise here. But what about fluctuations, right? What about fluctuations in cell size? To get a probability distribution that is nice looking, right? Maybe Gaussian, I don't know. So then you need to also talk about variance of the distribution, right? So mean is not sufficient to characterize the statistics of the system, right? So how do you compute variance? So variance, calculating variance is easy. You can just square this equation, right? They take a square on both sides and average it, right? So I define a new variable called S of n. And S of n is log of the cell size. And you can take a square of this on both sides. And what you find is that the square is given by S naught square plus kappa square, which is the growth rate, and the sum of variance of all these noise distributions, right? So one important fact here is that the noise is uncorrelated. So between each generation, the amount of noise you have in division time is uncorrelated, which is actually true. You can verify it from data. What you see here is that the variance in size is the sum of variance of the noise, right? And each of these noise distributions, right, has a finite variance. So at the end, we are left with a system where the variance actually grows with the number of generations, right? Where n is the number of generations. And 6 sigma square is the variance of each of the noise distributions, right? So, which means that the variance grows unbounded, right? So this is, by virtue of this model, you don't converge to any size. So ultimately, you know, the size distribution will be probably uniform, okay? So there is no size control here, okay, in this simple timer model. Okay, so the timer model is unstable, right? Which I think is a very profound result, right? In terms of, like, you think about a simple model that, okay, a cell doubles and then it divides, okay, this is what everyone thinks intuitively, right? But that model, if you do the math, shows that, you know, this is unstable, right? Okay, so so here is a model that didn't work. A model that works is something called the adder model. Okay, and this model came up about the time when I was doing my postdoc around 2014, around that time, and then a lot of studies followed this. The assumption of the adder model is that cell divides after you add a constant amount of size. Okay, so you're not multiplying a constant amount of size as in the timer model, but you add a fixed amount of size, right? So cell is born with the size V, it grows exponentially, and cell divides when a fixed amount of size delta is added, okay? So you increment by a constant size. So this is what's called an adder model. So literally you're adding a fixed amount. This model is very interesting, okay? So if I write the recursion relations, right? So VD at generation n is VB at generation n plus delta n, which is the amount of size that you add in generation n, right? In the next generation, what happens, you divide the cell size at division by half, and then you have this sort of nice looking recursion relation, right? Between cell size, between cell size of two different generations, right? What you can do simply is take a mean on both sides, okay? Because here, because the mean of VB at n plus 1 should be the mean of VB at n, because you're taking a mean about all generations, right? So when you take the mean, what you find is that the mean is just equal to the added size, right? So the cell size converges to the added size. So this is very interesting. So what it means is that if you grow exponentially, add a fixed amount of size in each generation, over time, what happens, your size becomes that added size, okay? So you converge to that added size, okay? I haven't shown what the variance is like, but I'm going to show that in a moment. But what this really means is that the mean is given by the added size. So if your size is larger or smaller than the mean, right? So sometimes errors could happen, right? The size, so there could be dispersion in sizes, right? So in biological systems or biological populations, right, there's a lot of variability in size and shape, right? Even amongst all animals. So in this specific case of this organism, by chance, if you end up with larger size, what happens is that by virtue of this rule, you'll converge to this added size within a few generations, right? So it leads to really fast convergence. And you can also compute the variance of the size, right? And the variance is bounded and is given by the variance of the added size, 1/3 times the variance of the added size, right? So it's a you get very well-defined distributions by virtue of this model. So this is called an adder model. And you can actually really verify this model from data. So what you can do is you can compute from the data that I showed you, like how much size you add in each generation, plot it against the bird size, and you see that it's constant. There is no correlation, right? So statistically, it's constant, right? So which means that, the ADAM model has been verified, not only in E. coli, right, which is the model organism I'm talking about, but in many different other organisms, okay, over the past years. And so this has emerged as a universal model, right, which is very rare in biology, but this is one of those models that's very universal, which came from simple phenomenological studies, like looking at data, trying out simple models, right? Okay. Now, so once you have this ADAM model, which gives you nice looking distributions, you can think about, so here is an illustration of what happens in the ADAM model. So here's a plot of the size on bottom left. I have a plot of size versus delta, which is the added size. And you see that through, if you're a small cell or a big cell, right, you converge to the mean size, which is the delta, through a series of divisions, right, by adding a fixed amount of delta. And this is actually shown using the experimental data here, right? So actually a bacteria adds a fixed amount of size, it converges to the mean size over time, okay? So now that you have the ADAM model, you can generalize it, okay? Is it the most general model you can write down where you have size control? The answer is no, so you can probably generalize it. And one simple generalization is this sort of linear function, okay? So you can say that the volume at division is a linear function of the volume at birth, right? You can probably write down more complex functions, but there is no reason to do so. So, the one thing I wanted to stress here is that we try to develop minimal models, right? So if you come up with a minimal model that describes the system very well, we stop there, but you can of course come up with more complex models, right? So this model is very interesting because it gives you several different, so it's a generalized version of the adder model and the timer model as well. So if A is equal to 1, you get an adder. If delta equal to 0, you get a timer model because you grow as a fixed multiple and you're growing exponentially. So that means that it's a timer, right? If A is equal to 0, it means that you divide at a critical size. Okay, so that's a sizer, right? And these different classes of models can be classified using this graph here, where I'm showing the difference between the cell size at division and birth plotted against the volume at birth. So these different models will show different correlations, right? So timer will show a positive correlation, like between the added size and the volume at birth. The adder will show no correlation, and the sizer will show negative correlation, okay? And so different organisms could do different things, which is captured by this general model, right? And in fact, if you look at different organisms, so here are some names of different types of organisms, some bacteria here, some yeast cells, which is, this last 3 are yeast cells. You see that most of the organisms are like close to adder, right? And sizer, like they lie within this quadrant between adder and sizer, right? Especially, we studied a lot about this special organism called Colobacter crescentus, which has this, very interesting geometry, which is curved, and I'll come back to it later about geometry and shape, which I think is very interesting. And it has a mixture of both timer and adder components, right? So different organisms can do different things, okay? And there is a lot of variability among species, but overall, this general model seems to work for most organisms, right? Any questions so far?
Shiladitya, hi, I had a question. So somehow the stochastic nature that you showed of the plot before, I mean, somehow in this adder model, it's not stochastic, right? Or maybe I misunderstood.
Yeah, I mean, I didn't explicitly say if it is, so what?
Because over time, the stochastic, the dynamics that you showed, yeah.
So stochasticity comes in the added size. That's why I have this label N. So the added size is stochastic, right? So it comes from a statistical distribution.
I see this delta comes from the distribution.
Yeah.
Okay. I see.
And the mean delta is the mean size. So what you see here is that the delta is not constant, but it's it has a statistical distribution, which is actually log normal distribution. I don't know if that's interesting or not, but it's not Gaussian. It has anyways.
Okay, thanks.
Yeah, thank you for the question. It's always good to get questions.
I had a quick question. Yeah, I was just wondering what the environment is in all these things. When you say universal,
yeah, that's a fantastic question, Narayan. So this the environment here is fixed, but I'm going to come back to what happens if you change environment in maybe 10-15 minutes from now.
But what is the environment actually?
Also, they're growing in nutrient medium, like rich nutrient, right? So glucose, for example. So you give glucose to the cells, they will grow, okay? So they need some sort of carbon source. And so they're growing in a fixed nutrient environment, which is glucose or maybe something else, glycerol. And this nutrient environment will modulate things like how fast you'll grow. So here, that is the medium.
But how about things like temperature and how about things like temperature and so on? I mean, is that
very good question? Yeah, so temperature.
Very
yes. So a very good question. So I'm not showing the temperature data here. It's published in this paper. But what temperature does is it slows down things. So if you reduce temperature, it slows down dynamics. And if you increase temperature, it speeds up dynamics. So temperature acts as a way to control the time scale, right? And yeah, that's what happens. But temperature doesn't have any effect on size and shape. What it does to this organism, if it's very cold, organisms will grow very slowly. It's very of course there is an optimal temperature. If temperature is too high, these cells will die. Okay, so usually between 17C and 37C is the range where 37C is too extreme for these guys. Okay, so
yeah, the word is a loaded word in that sense, right? Okay,
so you can change temperature, you can change nutrients, you can also apply antibiotics to this bacteria. I'm going to come back to it because antibiotics kill bacteria, which is what you want to do. And I'm going to come back to it. Okay, so
sure. Thanks.
I just want to talk about briefly. So this model is very phenomenological, right? So there are no molecules here. Okay, and if you talk to a biologist, they'll say, okay, I don't believe you. What happens at the scale of molecules, right? How does a cell sense size, right? It's not sensing any added size. There has to be some mechanism, right? And so, so cells actually sense concentration, okay? So they have no mechanism for sensing geometry. What they do is they can sense molecules, okay? And I'm going to give a simple molecular model for how this is achieved, okay? So let's imagine there are some molecules called division proteins, and these are labeled by these green dots here, right? And as the cell grows, the division proteins also grow because as you grow, you make more stuff, okay? Right? So you have more ribosomes that make more proteins, right? So the rule works like this. So cell grows exponentially, and this division proteins X, which is the number of this division proteins, grows in proportion to the volume. So if you have more volume, you can make more proteins, right? Which is and cell divides when you reach a threshold protein level, which is X naught, okay? It's very simple. So cells grow exponentially, the number of proteins grow in proportion to the cell size, and cell divides when you reach a threshold X naught, right? And if you do the math, it's very simple. I mean, I give it in my undergraduate class as a homework. So you can compute the added volume in each generation. And the added volume is given by the ratio of the two rates, K and Kp. K is the rate of growth, and Kp is the rate of synthesis of this division proteins, okay? And these are all fixed in a given environment, okay? So the question about environment is very important. So in a given environment, K over Kp is fixed, right? So the cell size is given by the ratio of these two rates, how fast you grow and how fast you divide, right? So the ratio of these two rates determines cell size, and you get a fixed volume, right? So here you see something very interesting. So a geometric feature is linked to some dynamic variables, right? How fast you grow and how fast you divide, right? So because these are rates, rate constants, okay? So what happens? So here's a very important figure, okay? So you can call it hypothesis at this stage. So a cell has a fixed amount of resources in its, in the interior of a cell. And using those resources, it can do different tasks. It can grow, it can divide, it can move, etc. Right? So it has to allocate resources between, let's say, growth and division. Like these are the two functions my cell is doing, right? This is a very simplified picture. And so if you spend more resources on division, which means Kp is higher, then you actually spend less resources on growth, right? So K is smaller. Okay, so there is a resource allocation constraint in the system. So if you spend more resources on growth, you spend less resources on division because you have to do two tasks, right, simultaneously. And this is true biologically because all the proteins are made from ribosomes, right? And the ribosome number is like your allocation, from which you do sort of optimize how to allocate resources. So you can see from this heuristic argument that if I spend more resources on growth than division, the size will get larger, right? But if I spend less resources on division than growth, the size will get smaller, right? And the volume is the ratio of these two rates. Actually, we verified this in experimental data, and you get a very nice linear correlation between these two rates. Since volume is the ratio of these two rates, what you will expect is that the volume, because Kp decreases with K, the volume will increase with the growth rate, right? So if you change the growth rate by changing nutrient condition, etc., the volume will increase, right? This is the first plot that I showed in my slides, and you can explain this using the simple principle of resource allocation, right? Yeah, so, and this is called the cell size law. But I call it a law because it was first discovered by looking at data, and about 60 years ago. About 60 years ago, people are studying, what is the relationship between cell size and growth rate, just by looking at data. This is the same sort of idea as in initial days when people did astronomy. They were just looking at data and try to look at relationship between variables, right? And the field sort of emerged like that. So you start looking at relationship. And here is the cell size law, which says that average cell size increases with growth rate exponential. Okay. Very interesting. And so now we have a theoretical model for how it happens, more detailed math, etc., which I'm not showing here, but these are interesting. Okay. Cool. So this is about size. So one interesting fact is that if you look into the living world, the bacterial world, it comes with a wide diversity of shapes, okay, geometry. And it's kind of fascinating, the amount of diversity you see in morphology. So you can, of course, get this simple rod-shaped bacteria, which we're talking about, E. coli. You can get curved bacteria, you can get sort of helical, so different types of geometry. And I've been wondering, are there like selective values like of these shapes? Like why do certain shapes have certain shapes evolve, right? Are this like adaptive evolution? So you can ask these questions, right? So then about a few years ago, what my group decided, we started collecting data for shape and geometry. And we wanted to look at if there are some universal laws of cell shape, right? So we already know like the size law, the growth law, etc., which I talked about. But there's some universal principles of cell shape. So here's my postdoc, Nicola, who's now a faculty at Queen Mary's University of London, recently became. So Nicola started looking at data for cell shape. And so we looked at specifically E. coli cells first, okay? I'm gonna show other organisms. And what I'm plotting here is surface area of the E. coli cells versus volume. And how do I get this data? So you can get images and you can do image analysis and you can compute these things, right? And So there are about 5,000 data points here. Each data point is in a different condition. So you have done different things to the bacteria. You have changed nutrients, you have knocked out some genes, you have treated them with antibiotics. So you've done a lot of things to this bacteria. And by doing these perturbations as shown here, the details are not important, but what you see here is that you can change volume over 2 orders of magnitude for this bacteria and surface area, right? And when we plotted the surface area versus volume, there was a surprise that all data collapsed onto a very interesting geometric relationship, okay? Which is surface area is 2 pi, volume to the power 2/3, right? And these are, so you expect that surface area scales with volume to the power 2/3 for, spheres, right? But these were sort of rod-shaped cells, which can be curved. Sometimes the shape could be really disorganized and disordered. But what was surprising is that when you fit the data and you find this very interesting relation. This prefactor 2 pi, so we saw that the prefactor was like 6.28, so we put a 2 pi here, okay? But I don't know what's the geometric origin of this, right? Right? So what does that mean, first of all? So that's something you can ask. So if you have a cylindrical cell, which most of the cells are, right? So if the surface scales as volume to the power 2/3, that means you're conserving the aspect ratio of the cells, okay? So that means that you grow, when you change their size, they grow in the same proportion, right? And this 2 pi is related to the aspect ratio. I'm going to come back to it, like what is the aspect ratio? So this is for E. coli, right? Volume to the power 2/3. We looked at the different organism, bacillus, like they're also another type of bacteria. There the scaling was 0.85, so which is not 2/3, but something else. But you start seeing this as nice scaling laws, okay, which is all what physicists like. And yeah, so this is for bacillus. So we were not satisfied there. We wanted to we looked at 50 other organisms, okay? So these are different types of archaea. Archaea is also another type of species. And you also see, like, they all sort of collapse under this power law scaling, right? The Staphylococcus is a bacteria which is round shaped. Yeah, it also falls on that scaling law, right? So this power law scaling, okay? So the idea is that so many organisms, so almost all organisms we look at have a power law scaling of surface area to volume, right? So the question for mathematicians is, what are the general solutions of shapes given this constraint, okay? You can, of course, work out what are the implications if you assume a shape, right? If I say, okay, my shape is like maybe a cylinder with hemispherical caps, which is most of the geometry of the cells are, but you can probably, I don't know, maybe some mathematicians can think like, what are the most general solutions of these classes of equations, right? Anyways, so here's some new math that came out from data analysis, but we can, like, as a simple-minded physicist, we can try to argue what happens at different scaling regimes. So if surface scales with volume, that means that the width of the bacteria is constant and you sort of grow in length, right? So surface scales as v to the power half, that means the length is constant, but you sort of grow in width. And if surface is v to the power 2/3, you maintain aspect ratio. Aspect ratio is constant, right? And these are true for, let's say, oops, what happened? Sorry about that. I think I lost my. Okay, give me 1 second. If there are any questions, I can take it now. I'll be back in a minute. My keynote crashed, sorry.
Does
Okay.
symmetry play any role in this?
Sorry?
Does symmetry play any role in the shapes?
Symmetry of the shapes themselves?
Yeah. I
Um,
mean, it's probably a physicist kind of question.
Yeah, so most of the cells are symmetric in terms of geometry, but they're asymmetric in terms of biological factors. But I imagine symmetry might play a role, but most of the shapes that you encounter are symmetric in nature. Maybe this type of cells are we haven't looked at the data, but we only looked at maybe symmetric cells which have definite symmetry axis. Okay, but that's an important question, right? Okay, so let's assume. Okay, so here is my. Oh, I'm not sharing this.
Yeah, you're not sharing it.
Okay, I'm back. Right?
Yeah.
So how is surface to volume regulated? So in general, we have this power law scaling. So S equal to mu v to the power gamma, right? And for simple shapes, you can work out what mu is, and mu is a sort of nonlinear function of aspect ratio. Okay. And if aspect ratio is 4, which you get it approximately as 2 pi, okay, which is for most bacteria, right? So previously we showed that the cell size increases with growth rate, right? And we explained that exponential relationship. So if you combine these two relationships, okay, you get something very interesting. So surface to volume ratio, which is maybe a fitness factor of this organism, you see that it decreases with growth rate, okay? So something like this. So surface to volume ratio decreases with growth rate. Now, why is that? Now, so if the cells are growing very fast, that means it has a lot of nutrients. And if cells are starved, it has less nutrients. Sorry, its growth rate is very small, right? So what does bacteria do is that they increase their surface to volume ratio when they are starved or in nutrient poor medium, okay? And that's an adaptive response. So you increase your surface to volume ratio so that you can increase the flux of food coming from the surface of the cells, okay? So by changing geometry and shape, what you can do is you can adapt to different stressful conditions, right? And this is how we interpret this equation, right? So different bacteria have different exponents, gamma, right? If gamma is 1, that bacteria cannot adapt to any condition. So that bacteria is probably very, but gamma is half or 2/3, they're highly adaptive. So for example, E. coli can adapt very well. B. subtilis, which has gamma 0.85, does not adapt very well to changing conditions, right? So surface to volume ratio decreases with growth rate. Very interesting. Now we can actually see it from the data. I mean, we don't have to, this is equation, we can verify it from experimental data. You can compute surface to volume ratio and you see that it goes down with growth rate, okay? Different organisms do it in different ways, but that's what happens, okay? Any questions? So, so the question is, how is surface to volume ratio regulated? I just wanted to give you this simple model for how to think about it. And this is a paper written by Harris and Terrio 2016. It's not our work, but the model for surface to volume ratio is also very similar to what we saw before. So the volume grows exponentially, right? So cells grow exponentially. And surface area grows in proportion to volume. The reason is that you have to make the surface of the cell, right? And those materials should come from inside of the cell. So the inside of the cell, you have this powerhouse where you build all the materials and then it goes to the surface, right? So the rate at which the surface will grow depends on the volume, right? And so you can solve this equation and you find that a steady state surface to volume ratio is given by the ratio of these two rates, beta over K, okay? Where beta is the rate at which surface grows, okay? So now we have almost a complete description of the geometry, like how surface area grows, how volume grows, how length grows, and how growth rate and division proteins are regulated. So using that, you can actually do different types of simulations, right? And I'm going to come back to some other, some simulations that we did with this. So there was a question about environment, right? So if you increase the amount of nutrient concentration, like if you feed the bacteria more, right? The volume gets larger, but surface to volume ratio goes down. And if you decrease the nutrients available, like less food, surface to volume ratio increases, but the cell size becomes smaller, right? So these are adaptive responses of the cell, but just based on the geometry and morphology. Similarly, we thought about this hypothesis a few years ago, okay? So as you know, many of you take antibiotics to kill bacterial infection, right? And so what antibiotics do is that it kills cells, right? So it kills bacteria or makes them grow very slow, okay? So when you study bacteria, you have to think about antibiotics because that's your biomedical application, right? That's how we get grants, right? So we write this motivating that, okay, our work will be beneficial for studying something more health related, right? So what you realize is that the geometry of a cell can actually determine the response to antibiotics. So for example, if you reduce your surface to volume ratio, that means that you have less surface area and the drugs will not get in that easily, right? So by reducing surface to volume ratio, you reduce the influx of drugs. So what bacteria can do is that when you feed them with drugs, antibiotics, it can reduce their surface to volume ratio and dilute the amount of antibiotics, okay? So that it has more advantage, like to remain and to evade the action of antibiotics. So this can be a mode of antibiotic resistance in bacteria. So in fact, we looked at lots of different shapes. This is very complicated, but let's I think my PowerPoint is crashing because its figures are very. Okay. Yeah, so let's look at this. Oh my God.
It crashed, yeah.
I'm going to skip that slide because okay. So what I wanted to say is that, so if we look at bacteria, what happens is that we looked at different shapes on the antibiotics, they actually reduce their surface to volume ratio. So, by becoming more round shaped from a very elongated shape, right? And that has the advantage of reducing the flux of antibiotics, okay? And so as a last sort of study, as a last sort of study that we did, and what I wanted to say here today was, you can do this experiment. So this is something we did in my postdoc lab. So we can watch bacteria, how fast they grow and under antibiotic action, okay? And so let me describe the experiment to you. So what I'm plotting here on the left is this growth rate of the bacteria, and I apply a drug at generation 0. Okay, so generation 0 is some generation. And what the drug does is it reduces the growth rate of bacteria. So you apply a certain dose of the drug to the cell. And what you find is that the growth rate drops sharply after application of the drug, but over time it comes back, it recovers its growth rate. So this is a mechanism for resistance. So you apply the drug, the bacteria initially suffers from it, but over time it overcomes the action of the drug, right? So it recovers fully, okay? So that was like, so there must be some mechanism for avoiding that inhibition, right? We looked at the morphology of the cell. So something very strange happened. So the cells aside from reducing the surface to volume ratio, as I said, reducing surface to volume ratio gives you advantage of diluting the drugs, right? So the width of all the bacteria actually grow, like they become very big. But interestingly, the curvature of the cells also changed. So they became highly curved, okay? So something sort of, as you can see here, the bacteria almost become C-shaped. They become very curved in response to the antibiotic. So that means that there is a mechanical effect of this, okay? So aside from affecting the rate of growth, they affect the mechanics of the cells, right? So here we need to think about the mechanics, because shape is connected to mechanics and forces, right? So how the forces are distributed on the surface of the cell determines its mechanics, okay? So now there was a point to build a model for these living systems where you take into account mechanics and geometry. And the interplay of both of these will determine the adaptive response of the cell, okay? And this is what we did. So I'll sort of walk through briefly with the model. So over the past few years, we are working on a model to predict the time evolution of living systems, okay? And the idea is very simple. It's sort of inspired by classical mechanics, okay? In classical mechanics, what you do is that you look at a system, you talk about generalized coordinates, right? And you write down, the Hamilton's equation for those generalized coordinates and momenta, right? In a living system, things are more difficult because you don't have a well-defined Hamiltonian, right? The system is dissipative, it dissipates energy. The system is also out of equilibrium, right? But we sort of found a way to work with that and sort of develop a theoretical framework to predict the time evolution of the generalized coordinates of the system, right? So if I look at a bacteria like this, right, I can decompose the system into some generalized coordinates which are defined by its shape, right? So Q is the generalized coordinates. So the shape is defined by the curvature of the cell, okay? The width of the cell, right? How wide they are, which is also varying along the bacteria, and also the length of the cell, right? How long they are. And you can write down equation of motion, right? For each of these bacteria. So it's, I don't have the time to go through this, like how do we get this sort of logarithmic form here? So, but what essentially is that the time derivative of Q is proportional to the gradient of a sort of energy functional E. And the energy functional will take into account growth, right? Because you're growing the system. The mechanics, sort of the elastic energy of the cell surface, the bending energy, and those sort of mechanical energy. So it has to take into account mechanical energy and growth energy. And so what you do is that based on this, you evolve the system. So the system has three components: the mechanical component, which is defined by this equation of motion that determines the shape of the system. There's a chemical component where the growth happens at specific sites along those systems. And there is a decision-making component because living systems make decisions, right? So, and which are very probabilistic. So as I said before, there is a probability of dividing based on the adder model, for example, right? So you couple these three components: mechanical, chemical, and decision-making, like stochastic. And then you have a complete framework of evolving the system over time, right? And then we can, so these dashed lines are theoretical models fitted to experimental data. So we can actually predict how, not only the rate of growth, but how the curvature changes, how the cell geometry, like the width of the bacteria changes, how the division time changes and all that, right? And so these are actually simulations of bacteria under antibiotic stress, like a single step. So what you see here is that the shape is changing, right? The shape is changing in response to the drug, right? Now that's a mechanical effect, and mechanical effect should be reversible, right? So instead of applying a single step-like pulse, what you can do is you can do an experiment where you apply a pulsatile drug, Okay, so this is similar to you taking antibiotic after every certain time interval, right? And what you see is that in that case, the shape is oscillating, right? The shape is, the curvature is increasing, then decreasing. The width of the bacteria is increasing and then decreasing. So, because when the drugs are not present, you come back to your original state. So it's a completely reversible process, okay? And that amount of flexibility actually gives the bacteria a lot of ability to evade the action of this stressful environment and stressful drugs, okay? Okay. So that's where I wanted to end today. And the sort of message I wanted to convey is that using simple mathematical models and physical models, you can learn or contribute a lot to biological systems, okay? And understanding the physiology and also sort of dynamic properties of living systems, which are very difficult, but you can start with simple assumptions, looking at experimental data, and try to test your model with data. And yeah, so I just wanted to thank my students and postdoc who did most of the work here, and I'm just a spokesperson. And at Carnegie Mellon, we have, I'm looking for PhD and postdocs who would be interested in, applying mathematics and physics to study living systems. Okay, so thank you for your attention. I'll take any questions.
So let's thank Shiladitya first for a fantastic talk. And we have at least one raised hand. Anand, you can unmute.
Oh, yeah, I really enjoyed the talk, Professor. Yeah, thanks. I had a couple of questions actually. The first one was about how, like if let's say 2 or more bacteria have a, follow a similar power law for like the surface to volume, and let's say with a similar exponent, how much variation do you tend to see in the kinds of shapes that they take? Like, not and because if the shapes are similar, maybe could there be a way to like formulate some sort of action principle from which you can derive the power law for the bacteria or something like that, some very general, or do we see a lot of variation in the shapes?
So we definitely do see a lot of variation in shapes because there are almost like 2 orders of magnitude in volume and surface area, which is a lot for these tiny cells, right? These tiny cells, if you 10 micron cube is not like the shape of the size. Bacteria are like sub-micron, right? So sort of microns. So we do see a lot of variation in shape, but the shapes could be very irregular, not ideal, cylindrical or spherical. It could be quite irregular. So you can approximate it as some self-similarity, right? So the shapes could be self-similar because of this. I don't know what you mean by action principle.
No, the reason I asked this was because you said that like, like those like mathematicians try to come up with a way to see if there's a unique, let's say, solution to the shape of the cell that satisfies a law. So like, what do we look at for that unique solution? Like, what kind of equation would we try to?
I didn't actually know the answer to that question. So that was a question to you. So my question was, given this power law or this equation, can you predict all the possible morphologies, right? And I think that's an open question. I can get some solutions if I assume some sort of, geometry of this, right? But I don't really have an answer to what are the most general solutions. So I think that could be an open question. And so I just wanted to highlight one thing. So some new mathematical laws that are coming out from just from data. So here is a lot of scope to work, I think. So in summary, I don't know. I don't have an answer to how do you proceed, but yeah, I mean, you can think about some sort of optimization principle that can give rise to some sort of shape constraint. And you have to come up with some optimization principle. Like, what is the cell optimizer, right?
Yeah, that's something along there. I was wondering if maybe there were ideas about how to do that yet.
So energy minimization is like one of the arguments in physics, right? So you minimize energy. So that's like what equilibrium physics is based on. But probably these living systems don't obey those type of principles. So it has to be something else, right? But it's clear, yeah.
Ramdas and then Narayan.
Yeah, okay. Yeah, hello.
Yeah, we can hear.
Yeah, okay. So what are the assumptions about cell mutations here? I mean, after generations, there are mutations that can happen. The
Yeah.
other parameters like environmental parameters like the growth medium and temperature we can control, but what are the assumptions about the mutations?
So we don't really see mutations occurring here in this constant, like if you do the experiment very long, so we don't see experiments, mutations here in constant environment within 1 or 2 days, okay?
Okay.
They might be happening. We did not do a sort of a genetic profile of this system over time. So I can't really answer, but even if these mutations are occurring, they're not really important for the type of dynamics we are, because the dynamics is not really changing in steady state, right? Under stress, probably some mutations happen, okay? And that those mutations can give rise to geometric changes, like, different mutations can give rise to different shapes. So we haven't actually studied those mutations in detail.
Yeah.
What we look at is actually what's happening at the scale of the cell, morphology, dynamics. We don't actually look inside. Okay. So maybe someone from microbiology, genetics background would be interested in that. And
Yeah. Okay.
Great question.
Narayan. Thanks.
Hi, Shiladitya. So one more question, sorry. So in all the pictures that you showed, it looked like division happens in the middle or something, right?
Yeah.
Is that universally true? So for example, you had this page where you had some Y-shaped molecule, for example, or something, and it would seem that it's more efficient maybe to have the constriction happen from one entity to 3 entities. Does that ever happen?
So 1 to 3 really happen. Yeah. So are you asking if a cell is dividing at one place or multiple? Can it divide at multiple places?
For instance, yeah.
Yeah. So it happens. So sometimes what you can do is some cells due to mutations can skip a division event and they can get very long. Okay. And those cells sort of divide at multiple places. Okay.
At the same time.
They will suddenly, so they can get very long and then they will tuck, divide. Right? Division need not be symmetric. Like this bacteria, for example, divides asymmetrically, as you can see.
Yeah.
Okay. So division, of course, can be, it's not 50-50 always. It can be 60-40, 70-30. And asymmetric division is something we are working on. We have worked on a lot. But multiple divisions can occur, but those are very rare events, like statistically rare events.
Wouldn't that also depend on the shape? Right. I mean, so that's the other reason I was asking earlier about the shape and so on. So it looks like shape could probably play a fairly central role in many things, right? I mean, is that, I have no sense of how this actually plays out in all this. I mean,
yeah, I mean, most organisms we have studied are rod shaped, like they have a long axis. Right. In most cases, as I said, what you said is true.
Yeah, so if it's a rod shaping, for example, it's sort of, it seems reasonable that it divides somewhere in the middle. I mean, equitable distribution of resources, whatever. I mean, something of the kind. But I mean, if it's
are you asking whether it divides in the middle or not? Does that depend on this? Yes, definitely. Yeah. Yes.
I mean, I think the shape probably plays more roles, but I mean, in particular, in terms of the location of division and so on.
Yeah, so you do have eukaryotic organisms that are like this, and they sort of divide, like dumbbell shape, like more kind of, and they could divide elsewhere. And the daughter cell could be significantly smaller than the mother cell. And
right, yeah, I see. Okay, thanks.
Okay, any other questions? I mean, Shiladitya, I had a question. So the final mechanical model that you proposed, with the generalized coordinates, I mean, I don't know if it's a relevant question, but like it was a first order differential equation. Is that an assumption or, you know?
Oh, that's, yeah. So, these systems are all non-inertial. And that's something I didn't say before. And the reason is that, so you don't do second derivative of time, is that mass is effectively zero because these are dominated by drag.
Okay.
So these are drag dominated.
I see. Thanks. I see.
So viscosity is so there is something called Reynolds number, right? Which is quite often used in fluid mechanics, which is the ratio of inertial forces to viscous forces. Okay. And in this system, the Reynolds number is of the order of 10 to the power minus 5. So you can safely ignore mass. So that's why all the equations are first order. But since they are coupled, right?
Yeah.
First order equations, so you see interesting nonlinear dynamics.
Right. Yeah, that's, yeah. And yeah, maybe just one more small question. So there was an equation where you had the equation for the volume and then the equation for the forget now which was for the growth. Is it? I mean, there was a there was an assumption somewhere that the resource allocation. No, maybe after this. I mean, resource allocation was like equally distributed inside the cell. I thought there was some. I mean, is that an assumption or is that what is expected?
It's a simple way of saying what I wanted to say, but it doesn't have to be equal. So let's say that there are some resources here, and you want to distribute into two tasks, right? And the two tasks, one could be smaller, one could be bigger. But the point is that they are coming from the same source, right? So all the proteins in the cell, which are these blue circles, and there could be different types of them. They are made by something called RNA, right? RNA ribosomes. That's the one piece of biology I'll just throw. And so the ribosomes are sort of limited in the cell. So then you have to sort of allocate resources between different tasks. Okay. So if you allocate one more resource to growth, the cells will grow and become bigger and less to division, right? So that was the argument I said. And so, so there is resource optimization going on, like some economics, like cellular economics going on, right? So you have, and that's also a very interesting field, right? How do you allocate cellular resources? And to do that, you need to optimize something, right? So this sort of, when you have trade-off between something and something else, there is some optimization happening. And I think so. These are very interesting questions.
Okay, thanks. Any other questions? I don't see any hands. Okay, in that case, let's thank Shiladitya once again for a fantastic talk.
Thank you so much.
Thanks a lot.
Thank you. Bye.
Thank you, Shiladitya. Bye.
Very nice talk.
Very nice talk. Yeah. Bye.
Yeah,
Bye.
thank you.